Okay. Ava, how's it going? What do you know about exercise physiology? If I say exercise physiology, what pops into your head? Okay, Don, you want to help her out? Everybody's on the spot. Hence the name tag. Especially during exercise. How the body works during exercise. Okay? Claire, stand up for us, please. Can you do a few jumping jacks? Yeah. Would you mind? Yes. You're welcome to come up here. That's even better. Okay.
How many do you want?
Just go and we'll tell you to stop. Emma, what's happening? Is she exercising? Nope, she's not exercising, is she? Yes, she is. Okay. Let's keep going. I'm sorry. As Claire does jumping jacks, what physiological processes are changing? Give me one. Excellent. Emily, can you give me another one? Claire, you want to help her out? What's changing? What does it feel like?
Sweaty.
You're sweating, okay? Why are you sweating? Do you move and not get sweaty?
Yeah.
Okay. Why do we sweat? Thank you. You can go sit down. We're gonna do this. For those of you that haven't had me, we do this a lot. Okay. If you won't talk and you won't answer, we will do squats until you talk. Okay. It's Phys. Why was Claire feeling sweaty? Why did her body temperature go up, Riley? You're 100% right. Why does exercise make your body temperature go up? Okay, yes. Okay. Generating that energy also generates what? Oh, you learned it in biochemistry. Why, when you exercise? Why, when you're cold, do you shiver? Make heat, right? Metabolic equation. Take glucose, oxygen, we make CO2, ATP, and heat. And that's why Claire sweats. That's thermoregulation, exercise in the heat. We'll talk about that. Her heart rate changed, right? Her muscles had to contract. Did your breathing change, Claire? Mm-hmm. Yeah. Her breathing changes. Her heart rate changes. Her stroke volume changes. Her cardiac output changes. All of these things have to happen just to let you exercise. That was just some jumping jacks, right? Now imagine we're going to CrossFit today, right? We're going to go over to F45 and whatever they're doing over there, make people do stuff, okay? That's what we're going to learn in class. In some instances, excruciating detail. Excruciating detail. What are the processes? What's the underlying physiology of each individual system that has to change to let us exercise? Where do we get energy? Why do we need that energy? What systems have to change their function in order to support all of that? If we understand that, then you can understand why some people can run farther than others or faster than others, or why you can lift more weight, I guess, than someone else. Okay? Or why it sucks to be old like me. Okay. So, you guys may not have ever thought about this, but in general, as a discipline, exercise physiology, we're very young. Very, very young. When I was an undergraduate, we did not have a program in this where I went to school. To be honest, we had 1,000 students. They still don't really have a program. They at least teach exercise phys. Okay. I didn't know what this was until after I had graduated college. That was in 2000. That's not that long ago. Okay. Not that long ago. But if we look at exercise physiology as a discipline, it has really come about in the last, say, 100 years or so. It's grown out of the ideas of anatomy and physiology. We're going to do a fair amount of anatomy today. We'll do some physiology today. Probably won't do a lot of exercise as it relates to that today. Remember, anatomy is structure, right? Muscles, bones, tendons, ligaments, joints, heart, nerves, blood vessels, et cetera, right? All the things that Cindy taught you or should have taught you. and then physiology. Physiology is about function, processes, function, pathways, signals, okay? Anatomy is structure, physiology is function. Exercise physiology really is how structure and function get applied to allow us to move, okay? How do they get applied to let us move? And then if you move a lot, are there going to be adaptations and changes? Okay. So if we look back in the history books, and I guess I should say it, at the very, very end, there's also, which I know you guys can't see here, this idea of what's called environmental physiology. And so environmental physiology and sport physiology, in some ways, are really subdisciplines of exercise physiology. Environmental physiology is it's hot outside, I'm sweating. Why does it feel worse when I go to exercise? Why does this reduce my? Okay. I went and I tried to walk up to the top of Pikes Peak. Anybody done that? Go to Colorado, go to Rocky Mountain National Park, go for a hike, and you're like, why in the hell is this so hard? Right? Altitude. Okay. Why do half the people that try to climb Mount Everest die? Altitude. Okay. That's environmental physiology. Really, really important. And then kind of a subdiscipline that can have some of this and ex-phys is sports or performance-related exercise physiology. What's the physiology of human performance? Why are some people better distance runners? Why are some people better sprinters? What are the adaptations to training that allow us to perform in certain particular ways? Okay. We will touch on these a little bit, but we're going to spend a lot of our time kind of here strictly on how exercise and the application of a single bout or chronic bouts of exercise change anatomy and physiology. Okay. This thing going to go? Maybe. Okay. Now, I said that this is a young discipline, and in a lot of ways, the way we approach it, it is. But if you look back in history, you can see back that the Greeks understood some things about anatomy and physiology and sports and kind of the applications of those things together. There was a guy whose name was Galen, and he existed right around 100 AD or thereabouts, and he was a physician to the gladiators, which mostly meant that he dealt with scrapes and puncture wounds, and I got attacked by a bear and all of those kinds of things. But he had some first theories of anatomy and physiology. Okay. Encompassed in that would have been some ideas of exercise and sport, or what we might think of as kinesiology. In the sort of middle 1500s was an early anatomy textbook by Vesalius. Very descriptive, didn't really focus on function. Okay. Somebody that's new. Catherine, why do you think a early textbook in anatomy would be very descriptive rather than being able to actually elucidate functions? And remember, it's the 1500s. Okay.
They had to use more words rather than like videos.
Okay, sure. Right. If I say, look at Bailey, can you describe her anatomy? Okay. But it would be very descriptive. You could say, like, okay, she has this muscle and it's here and it looks this way, but I can't say in her cells, right, her mitochondria are doing this. They didn't have microscopes or anything else, and they couldn't actually see inside other than you were dead and we were going to dissect the cadaver. But again, that's very, very descriptive, very big picture. But we're beginning to get sort of kind of to that place. Okay, we're beginning to get there. So as we evolve kind of over time, one of the very first true textbooks, right, on physiology, and that you might call exercise physiology, was in the late 1800s, in about 1890. The origins of exercise physiology are all about muscle metabolism. Muscle metabolism. Okay? Using energy. Where do we get the energy? How much of it are we using? Why do we use more or less in certain circumstances? Okay? in 1900 or thereabouts, a guy named John Haldane developed the first O2 sensor and O2 analyzer. I know from your previous classes, you guys have talked about oxygen consumption. This is the way that we measure without looking into your muscles. We can measure your muscle metabolism, analyzing the amount of oxygen that you're breathing in and using to make ATP in your muscles. So that only began a little over 100 and about 125 years ago. Okay, he developed something called a Haldane analyzer. Back in the dark ages, when I was a grad student in 2001, we had to use a Haldane analyzer to look at oxygen consumption in one of our graduate courses. We had to figure out how they did it in the old days, and then they let us use what you guys have available. We have available to us now. It made us very appreciative. 1921, a guy named A.V. Hill won a Nobel Prize by looking at muscle metabolism. The first, and in some ways, sort of the only Nobel Prize winner in exercise physiology. Okay? 1921. He was one of the first people to do studies on runners. Some of the things that he was looking at in muscle metabolism in animals we're still trying to figure out and repeat today. Okay? 100 years later, and we're still asking the same kinds of questions. So, this is going to begin, as we're going to move from animals into humans, this is going to begin to give us some ideas of, it's going to start looking like modern day exercise physiology. Probably the seminal or most important moment in modern exercise physiology was the founding of something called the Harvard Fatigue Lab in 1927. The Harvard Fatigue Lab was founded, guess what? Harvard, right? But it was in the business college. In the business college. You may think, why in the hell is it in the business college? Well, in 1927, there wasn't OSHA. Okay? There really weren't unions that much. So what they were very concerned about was working conditions in factories. Okay? And this was, what are the optimum working conditions to get people to work longer and work more efficiently so we can have better production and make money? Let's not think that they were really kind of out to try to make things better. But they were interested in what they were calling fatigue, or the inability to continue working at a certain rate, right, over time. It wasn't air conditioning in 19. Okay? There wasn't a lot of heating in 1927. And so they were very concerned about those sorts. It was directed by a guy named D.B. Dill, and it brought in sort of world-renowned scientists from around the world. Okay? They looked at the physiology of movement and the effects of environmental stresses, heat, humidity, altitude, and things on exercise and work. So from 1927 until about 1945, when it closed at the end of World War II, there were about 350 articles that were published from their group. Nearly every modern day exercise physiologist can trace their training in some way back to people that came out of the Harvard lab. Closed. All of the scientists went off back to their home countries and back to other places, and then began their own versions of the lab. And that is really what has sort of been the founding of some of these kinds of things. Okay. So for instance, I have taught you guys, or I will have taught you guys. One of the guys that taught me at Georgia was taught by his father. And his father was taught by D.B. Dill. Okay? And so you can look back at sort of like 6 degrees of separation from the Harvard Fatigue Lab in those ways. But this was the sort of founding event that set the seeds for all of our modern exercise physiology or kinesiology sort of departments and laboratories. Okay? So here they are sitting around in their chairs. They're outside. They're breathing into these kinds of things. This is their ability to look at and measure O2 and CO2 use and production. You'll note they're sitting in like their suits and in jackets and things. They were outside at altitude. But we do basically the exact same things. They would breathe into these big bags. They're called Douglas bags. It looks like this. And then you collect all the gas, and then you go off and you run them through the analyzers. So we do the same things now, but we just run it directly into something where we can analyze it. Okay. So from the 40s until the 70s, a lot of the kind of advances were made in Scandinavia. Okay? One of the reasons things were done there is their people are sort of down for anything science, right? And I will talk about, but it's a lot of things that happen are the idea of taking muscle biopsies, the idea as a culture of we're outside, we exercise, we care about these kinds of things, right? And so there were a lot of kind of big advancements in sort of the Scandinavia. And one of the biggest ones was this idea of what's called the Bergstrom biopsy, which we'll talk a little bit more about later today, I imagine. But here they are taking a piece of muscle tissue out from the vastus lateralis. And then from the biopsy, we were then able to look inside of muscles. We've gone, as we were talking with Catherine, about observation, looking, seeing, describing to now I can see inside. Okay. What does it look like on a cellular level? How are those things changing? Okay. And then we've now advanced into, so this is a light microscope look at a muscle biopsy. Here, what you can see here is an electron micrograph of these are going to be individual sort of muscle fibers and myofibrils that we can see here. I saw an even greater advance of thing the other day from something called the fusion tensor MRI. This is really crazy. They can measure blood flow and stuff, and they can use the blood flow patterns and then recreate the underlying tissues. And you could see all of the muscles in your quadriceps and like all of like the individual like muscle fibers in there in the directions they were all running just from an MRI, which is pretty wild to me. Okay. Things that we will go through in class, I'll try to point them out. We have learned from these advances, things that we know about fiber types, about mitochondria in muscles, about changes in capillaries and things in muscles, glycogen and glucose use, fat storage in muscles have come from our ability to take biopsies and look inside of the muscles. Okay. In the US, there were two kind of driving events. They're all based off the Cold War. You guys know what the Cold War is? For a while there, the last time that I had to teach this, right, we'd had the Cold War and it was over, and we're like, the Soviets and Russia is not really our enemy. They've kind of come back to be our enemy in those ways, right? But wanting to go to the moon, and then to beat the Russians, and then wanting to beat the Russians and win more medals at the Olympics were kind of the driving factors in sort of advances in exercise physiology in the US. We'll talk about we needed to understand microgravity and changes in physiology that are going to take place with space flight to let us go to the moon. So there was lots of money because of that. And then in the early 70s, late 60s, early 70s, we started getting to the place where we want to win more medals at the Olympics. We need to understand the best ways to train, what's going to make people better distance runners, middle distance runners, or sprinters. And so again, money. Okay? Research is driven by money. The government has most of the money. They make the decisions on what they prioritize. And so in this instances, they were prioritizing these things. So there was money that was available. So, all right. What about things today? What do we do today? And I should have updated some of my pictures, but modern exercise physiology, you guys have available to you from your watch, right, or a heart rate monitor, information that even 10 years ago we didn't have, right? You can go for a run or a bike ride and know your heart rate for every moment that you were out there and have it downloaded. You can track your sleep. You can track your heart rate variability. You can know exactly how far you went. You can know what your pace was, right? You can watch your pace as you're going. You can monitor all of these things in real time. And we couldn't do that even like 5 or 10 years ago. Okay. I have no idea what the next like 5 years is going to be. Something like pop up in my glasses or something, right? Like just put a contact lens in and it's going to display stuff, right? And then the machines take over. But we have this kind of quick and easy fitness test. We can estimate your VO2 max from walking test or from just going to run, right? And be relatively accurate about all of that. You can figure out your use of carbohydrates and fat during exercise now. We'll talk about a thing called RER. You can buy stuff. I see it advertised on Facebook and other social media where you like blow into this thing and it measures what's called your RER. And it will tell you, like, are you on basis or not? Or what are you using carbs? Are you using fat in those things? There's been a big industry on supplements, right? Supplements, testing supplements, ergogenic aids, sports drinks, protein powder, all the stuff the bros at the gym are using. Okay. And so that's a lot of the stuff that we do now. We've also gotten into big deals about exercise and genetics. Yesterday was Mother's Day. Did you thank your mom for your mitochondria? I'm serious. Whatever mitochondria you have, if you are a good endurance athlete, it's because you have good mitochondria. You got those from your mom, right? Genetically, mitochondria come from your mom. We're trying to get into and understand the genetics of exercise. Some people are have better adaptations to training, and it's genetic, right? Some of you can run all you want, your VO2 max will never ever go up. I'm sorry. Okay, I'm sorry. But that's becoming a big deal. And then this idea, a lot of the stuff that we cover in health and fitness is the idea that exercise is medicine is also the big push, right? This is why you guys take health promotion class. We know if you exercise, it's good for you, and yet we still won't do it. I'll speak for myself. I know it's good for me. I still won't do it. Right? It's hard. I don't know how to change behavior. I mean, I do, but you know, if someone wants to like take care of my daughter for like 2 hours a night so I can exercise, that'd be great. Like you guys can walk the dog and fix dinner and all of that sort of stuff. That'd be awesome. But that's kind of where this is going. Okay. Where are we going in the future? It's going to be more about the interaction of genetics, exercise, medicine, kind of health outcomes, changing behaviors, getting real-time feedback on all of these things. Okay. What am I doing on time? Okay. So before we jump into some other things, actual kind of anatomy, I want to introduce two concepts to you guys. They're going to come up over and over and over and over again in class. Okay. That's the idea of what we call an acute response and a chronic adaptation. Okay. An acute response was what we did with Claire. Stand up, exercise, what changes? Acute means short term. What changes right now? Okay. Right now. That's an acute response. If you're going to be able to exercise, there are certain acute responses that must happen. If they don't, you can't exercise. You certainly can't exercise for very long. Okay? What happens when we get an acute response is when you exercise, you throw your physiology out of homeostasis. Right? You remember what homeostasis is? Yeah. That's right, a balance, right? Balance. When you exercise, you take yourself out of homeostasis. Okay? Or you try to. And that getting out of homeostasis is one of the things that the body doesn't like. And so you will then get chronic adaptations if you exercise a bunch, and the adaptations are going to occur to limit the disruption of homeostasis from an acute bout. So an acute response is you do something now, what changes? Heart rate goes up, blood pressure goes up, et cetera, et cetera. A chronic adaptation is my muscles get bigger. My muscles get stronger. My heart gets bigger. My blood pressure goes down, right? And so these chronic adaptations are usually what we're going to call training adaptations. And these are the things that are going to improve health, improve exercise performance, right? Sport performance. These are relatively permanent-ish types of things. Change in anatomy, change in actual physiology, changes in structures that are going to limit then the disruption of homeostasis the next time we have Claire from the If Claire did jumping jacks for 2 minutes at the start of every class, maybe by the end of our time together, she'd have some sort of adaptation. Or you want to do that every day? Like every day we'll give you like some more weights or something. You can do jumping jacks with all of that. But that's the general idea. Okay. Do you all understand that? Does that make sense? Yes? Okay. Okay. So I just want you guys to think about this for a second. And I'm going to try to embarrass you like it's the first day. So, how and why is exercise physiology important to you? Okay. And what role do you see it playing in whatever career you're going to go after? Right? You're in our major for some reason. This is an important class, right, in our major. So why do you think this is important? Because I always ask this because I think you need to have some level of ownership. You have to understand, I am here, right? We have this class, not always in the best sequence, but it's part of our major for a reason. We think it's important. We need you guys to understand why it's important as well. Or at least I need to try to understand why it's important to you. So that then I can have you take some ownership on here's why you should learn the Krebs cycle. You shouldn't. There's no. Right? I've had 2 semesters of organic chemistry. Never used it before. Never. Yuck. Terrible. Okay. Krebs cycle, less, but we do a little bit of this. Let me ask this. All right. Kelly, why is exercise physiology important to you? On the spot here. You like to exercise. Okay. Okay. Can you guys done that? You guys learned anything in any of your classes up to this point and actually change how you exercise train? Okay, good. See, we've been mildly successful at actually changing behavior. That's good. Somebody else. Adam, what about you?
I've been specialized in like sports medicine and orthopedics.
Okay.
So I think understanding like causes of certain injuries really helped me and like diagnosing.
Perfect. See, very important. On the back, Colin, who's a Padres fan, or at least is wearing a Padres hat.
Well, I want to go be a PA, but I
Okay.
also work for a.
Sure.
So need to know how this why does this that.
Okay. Great. I would argue every medical professional needs to know some exercise. Right? Exercise is the single best medicine that we can do. And you go to the doctor's office and like, I don't know, you should exercise. And like, what should I do? Like, I don't know. They don't tell you, right? They don't know anything. So when I go to the doctor, I'm hard on doctors. I have very high expectations for what they should be. They should be smarter than me. Smarter than what I can do. So I think that's important. Okay, we'll just kind of keep going around. Maddie.
Me?
Oh, I'm sorry, there's multiple. So you're both kind of in the same line. It's like, sure, Maddie Brown, you can go first, and then we'll let Maddie Ward go.
Both questions.
Whichever one you'd like to answer.
Because everything in I think occurs at rest and during activity.
Okay.
And like the physiological mechanisms, blood pressure, heart rate,
Okay.
all these things occur in both instances. And not even exercise, but like disease is gonna impact that. So how can what we know about adaptations and responses from exercise be applied to disease and wellness? And I want to be a doctor, but like
No, that's great. I think that's 100. Right? You have to know how it works, or it's supposed to work under normal circumstances, right? Then be able to understand what's not working. Very soon, you guys will wake up one morning and be like, why does my back hurt? Why can I not walk today? Right? Why did I have 3 drinks and I can't function anymore? This didn't used to be a problem. That happened to me this week. On those kinds of things. Right? Why do my muscles not look like they used to? So again, personal, keep speaking from personal experience again. So it's important to understand this. Okay, good. Adding number 2, and you want to add,
I want to be a pharmacist.
okay?
So I think knowing what adaptations that you can use before you rely on medication,
Mhm.
is something that I want to implement into my life.
Okay. You gotta know how things work, but you know where the drugs are going, right? And what drugs to give people. Or how those drugs might interact with people that are going to exercise. Okay, very good. All right, we will take about a 5 or 6 minute break for you guys to get up, walk around, go to the bathroom, check your Instagram. It is social media break. Okay. Purple on dark blue on purple. Everything's looking great. So we are going to start in what I think is the most logical place. Other people seem to disagree with me. Dr. Campbell and I used to always argue about this. I think muscles are the most important sort of tissue in your body. Okay? That me. Muscles are the most important thing. Prove me wrong. Or whatever. But we're going to talk about muscles first. Okay? Without muscles, you can't move. Okay? Muscles are where metabolism takes place. So you use and make energy in the muscle. That lets you generate force, which lets you move. All of the other changes that take place that Claire experienced with her acute bout of exercise generally occur to support the fact that muscles are contracted. And so I think it's all kind of a natural consequence. Let's learn about our muscles. How do we move? How do we get force? The amount of force controls the amount of metabolism. We'll learn about metabolism. The amount of metabolism controls the cardiovascular response, which controls the pulmonary response. And then all of those things are going to control our adaptations to training. So we're going to start here. Okay. This is how it makes the most sense to me. So I'm going to try to explain it to you guys in that particular way. Okay. So we're going to go from sort of an anatomy standpoint first. Okay. You guys have probably seen this slide before. But muscles have one job. Okay. One job. Generate force. Okay. To generate force, they must use energy the vast majority of the time. Okay. Your metabolic rate, or your resting metabolic rate, is primarily determined by how much muscle you have. And that means that's how many Doritos you get to eat in a day. Okay. Really important things in life. Okay. Now, guys, stand up. Damn it. It's not in the back of the classroom. There's more space now. So tell us, play Simon Says. Dr. Black says, touch your nose. Okay. Dr. Black says, hop on one foot. Sure, yeah. Okay. Stop hopping. Dr. Black says, stop hopping. Okay. Now, thank you. Okay. How did you do that? When I said touch your nose, how did you touch your nose? Yes. I taught you something last semester, right? You thought about touch my nose, right? Even though ever so briefly, you thought about it. And then you didn't. Then I said, keep touching your nose and hop on one foot. Okay? Your brain sends signals to your muscle. It goes through what we're going to call, right, the neuromuscular system to do this. You have what are called motor neurons in your spinal cord. They're connected to the motor cortex in your brain, and they connect out to muscles, and they allow you the ability to, under most circumstances, only contract the muscles you want to contract when you want them. Okay? Now you have a cramp or something, you're going to get a contraction that you don't really want. It's not under your control. But in general, we have what is called voluntary control. Okay? You choose what to do. You choose the manner in which you do it. Okay? And all of this stems from the function, especially in your nervous system. So we'll talk about what are the structures, what are the processes, and how do we use all of that to let you guys write and type, right, or play the cello, or run, or go up and down stairs, or whatever it is that you're doing, very, very precise. Okay? And that's why muscles and motor neurons are important for exercise. They're important for all movement. We need to contract at times hundreds of muscles simultaneously to generate a very, very precise amount of force at a very precise instant in time so that you can do whatever it is that you want to do. Okay? Walking is an incredibly complicated thing. Okay? Just standing up and having balance is going to require tens of muscles at just the right amount of force production, as well as the ability to integrate things that you're seeing and feeling and sense of position to be able to control what's happening. Okay. Much less, I'm going to swing a golf club, right? Or you're going to play the guitar or something that's very, very neuromuscularly complicated. Okay. And so the reason we're going to make a big deal about all of this stuff is because the structure, the anatomy, and the physiology of the neuromuscular system all fit together really, really tightly to let us do the movements that we normally do in our everyday. Okay? If it was organized differently, if we were, let's just say, if we were quadrupeds instead of bipeds, our anatomy would be different, right? Your shoulders would look differently than they do. Okay? Your dog or your cat or a horse or a tiger or something has very different sort of anatomy and physiology, right? Animals that have to swim all the time, like fish, have different fiber types. They have different processes because the movements they need to do to live and survive are different. And so it's all evolved to be kind of where we are in the midst of this. Okay. Okay. Fun facts. There's a little over 650 muscles in your body. Okay. You get struck by lightning, they'll all contract simultaneously. That's not very fun. Okay. Muscles are made up of thousands and thousands of individual cells that we call a muscle fiber. Okay. A muscle cell is a muscle fiber. Muscle fibers are, if not the largest, I should have looked this up, they are one of the largest cells in your body. I could pull out one muscle fiber from your quadricep and you can see it with the naked eye. Okay? There are not many other cells that you can see without having to use a microscope. Cells are relatively cylindrical. We'll talk about those a little bit later. Because each muscle fiber is so big, or each muscle cell is so big, it has many nuclei. That becomes really important for our ability to make protein, neurotransmitters, and all of the things that muscle fiber needs to be able to function. Okay? That makes muscle cells unique. Most cells in your body have one nucleus. Muscle fibers have multiple. Okay? Fiber length is going to vary. A few millimeters for the small muscles in your eye to potentially up to like 30 centimeters in the large muscles in your legs, right? So like we could pull fibers out of your vastus lateralis and I can see them. Okay? Now, you got to let me cut your leg open and do a bunch of stuff to do that, but we could see them with the naked eye. Pretty crazy. Okay? Muscles are mostly water. They're really mostly water and protein. And the other 5% is phosphates and ions, right? So look around, I don't see too many people that have their giant like metal water bottles today. I will give you grief for those. My wife made me buy her one yesterday as a Mother's Day present. I just rolled my eyes. I'm like, what? What are we doing? Right? But apparently we'll keep her Mountain Dew cold for like 3 days, which is a really important thing for her on all of that. Muscles are mostly water and protein. We'll get into this a little bit later. Okay. The protein side of things becomes really, really important when you're thinking about making muscles get bigger and how they're going to adapt. Okay. I know you guys have all seen this before. I've made this analogy before. I think it's a fairly apt. Muscles are a Russian nesting doll. Guess what a Russian nesting doll is? Avery, remind us what a Russian nesting doll is. Okay. If we look at how muscles are organized, what you think of as whole muscle, bicep, let's just say, okay? Cut that open, cut through the connective tissue, the epimysium on the outside, and you'll see that muscles are made up of what we call fascicles, which are going to be these kind of longer tube-like things. You cut open the fascicle, and inside the fascicle, it's a collection of more tubes that are individual fibers or cells. You open the fiber up, and there's these little tubes inside the fiber that are called myofibrils. And then the myofibril is these little tubes that are stacked end to end to end to end on each other that are called sarcomeres. And the sarcomeres are made up of little strings or tubes, proteins, which are going to be actin and myosin are thick enough filaments. Okay. So in that particular way, it's just these increasingly smaller little cones or cylinders that are going to make up a muscle. We're going to spend most of our time talking about what happens in the fiber, the myofibrils, the sarcomeres, and the proteins that make up the thin and thick filaments. That's where a lot of the actual physiology is going to take place. But something's going to happen here at the protein level, which then is going to cause movement, which means the sarcomere moves, which means the myofibril moves, which means the fiber moves, the fascicle moves, and that is then how the muscle shortens or lengthens, and then it pulls on a bone and you get action. Okay. Okay. I do want you guys to know these sorts of things, because it's really, really important for our ability to move and our ability to train. There are 3 layers of connective tissue that are going to surround all of these tube within a tube within a tube structures in a muscle. Okay. Here is a muscle. You're going to have basically your skin, and then under that, there's what we call the epimysium, or you guys may have heard it called a fascia, or something like that. Okay. And so this is connective tissue that surrounds all of your fascicles. Okay. It surrounds all of the fascicles. So it encompasses sort of the entire muscle. That's the epimysium. The perimysium, it's the same thing. It's more connective tissue, more collagen-based stuff, and it surrounds each individual fascicle. That's the border around a fascicle. So you're gonna have sort of perimysium that surrounds our bundles of muscle fibers. And then around each muscle fiber is what's called the endomysium. And again, it's the same thing. What this does is it holds everything together, and connective tissue is like a rubber band. Okay. You pull, the muscle moves, there's going to be force. This stuff is kind of stretchy, and it lets us kind of pull and transmit force. And then when the force goes away, it is elastic and it goes back to its original shape. So this gives us some level of kind of structure, okay, to the organization of a muscle. Okay? So I do want you guys to know these individual. Okay? All right. I should also say that essentially at the end down here, all of this comes together, and the epimysium is going to pull down together, and it's going to connect to a tendon, right? So the muscle has a tendon on the end. The tendon connects that muscle to a bone, And so when we get a contraction all the way down on the sarcomere level, it's going to move, and all of that movement and force gets transmitted through the connective tissue and eventually gets transmitted to the tendon. And that is what gets put onto the bone, and that's what allows us to do. So without this connective tissue, we don't get force transmission and movement, and we lose some of the shape and structure, kind of organizing structure in our skeletal muscle. Okay, I talked about that. Did I not do the other? Okay, so we'll talk about this and I'll get the basement membrane here in a second. Okay, here's the inside of a muscle fiber. We'll go into quite a bit of detail about one of these individual structures that are going to be here, okay? Because they're all really, really, really important, right? Many of these are going to have sarco as their prefix. Sarco is Greek or Latin or one of those two things, and it means muscle, right? So the muscle membrane or the muscle fiber membrane is called the sarcolemma. We'll talk about the sarcolemma a lot, probably not today, but tomorrow. Right? It's really, really, really under the sarcolemma. We're going to have something called a basement membrane, which I'll show you guys some pictures of. You guys talk about the basement membrane at all in human physiology or in anatomy? No? Anybody ever seen a house without the walls up? Yeah. Okay. The basement membrane is, in essence, it's like the framing around where the walls are going to go. You put drywall up and stuff like in your house. So it's going to, again, sit on one side of the sarcolemma and give us some structure that's going to be there. And I mentioned the basement membrane because there's some proteins in there where we have some genetic mutations that it doesn't work very well, like muscular dystrophy. Dystrophin is a protein that's in the basement membrane. So if you get a mutation there, then your muscles are not going to do very well. So we'll talk a little bit about that. There are these structures, these tubule structures called transverse or T-tubules, which are the gold-colored things in this. We'll talk about their structure. The T-tubule is basically a tube of the sarcolemma that goes from the outside down to the inside of a muscle. They're from a muscle fiber. Really, really, really important to be able to bring an action potential from the sarcolemma down inside that fiber to get a contraction. The T-tubules are going to connect to these other tubular network called the sarcoplasmic reticulum, which is here in kind of dark red. What's in the sarcoplasmic reticulum that's important, Dustin? You remember? Yes, calcium. Very good. The SR has calcium in it. No calcium, no muscle contractions. Okay? You get calcium in a place it's not supposed to be, your cell kills itself. So calcium is really important. Keeping it in here when we want it to be in here, also really important. We'll go through the ins and outs of that. We have mitochondria. So mitochondria are inside of a muscle fiber. They're inside the muscle cell. Okay? Like the nuclei, there will be many, we hope, okay? Many mitochondria inside of a muscle fiber. The more mitochondria in the fiber, the more ATP that fiber can make through aerobic metabolism, which we will talk about next week. Okay, so mitochondria are really critically important. They're in the fiber because, as you guys will see in a few slides, we need the ATP on the level of the proteins, on the actin and myosin level here in a sarcomere. So we want the ATP producing structures to be very, very close to those kinds of things. So we don't have to bring the ATP very far to the place where it's going to need to be used. The last two things are going to look about the same under a microscope, but they have somewhat different functions, and that's the nucleus and a thing called a cytolysis. There are many nuclei in a muscle cell. Muscle nuclei look and function just like nuclei in all your cells. They have DNA, right? The DNA will make RNA. They make proteins. They make hormones. They make all the stuff that you need inside the muscle fiber are going to be made and derived by signals going to the nucleus and telling it to activate that particular gene, transcribe that protein, or that hormone, or that. So, in addition to our nuclei, which I should point out, Myonuclei are on the outside of a muscle cell. They exist. They're on here on the very, very outside. They're not down in the middle like most other cells. Why might that be? Anybody want to guess? Why have a nuclei on the outside rather than on the inside? Carly, you have any ideas? Nope. Okay. Michaela, can you help her out? Why the outside and not the inside? Nope. Okay. It's all right. Anybody? Zach.
Maybe like when you're like rebuilding the muscle after like you did it or like working it out.
Okay, that's an interesting idea. It's not exactly what I was thinking or what the textbooks would tell us, but that's a very reasonable supposition. Okay. What about the sarcolemma? You don't know, you're just like guessing it's by the sarcolemma?
You said it was like sarcolemma is important.
A good guess, okay? The real answer is they're out of the way. There. You can fill up all of the inside of the cell with actin and myosin with sarcomeres, okay, or with myofibrils. More protein on the inside means more force. That's the big important. By having the nuclei on the outside, right, they're not going to take up as much space on the inside. And that's going to be really, really important for us. Now, does anybody remember what a satellite cell is? We heard of a satellite cell. Rock. Yes, they're definitely involved in the regeneration process, right? Avery, you okay? Okay. That happens. I'm going to say, don't let it pinch you. We'll try that. Okay. Five. They're stem cells. That's right. Satellite cells are muscle stem cells. What does a stem cell do? I didn't remember. Maybe like transplant. Multiple uses if I work on an inserting.
Yeah, like
You're getting close.
the stem cells can't they like people turn it into like generate tissue and stuff.
Sure.
Then you like just put science stuff to it. I don't know the word, but
Okay. Danny's getting close. So stem cells, true what we call pluripotent stem cells, right? Are cells that have the ability to become anything, any other tissue in your body. Okay. All of you began as two cells. Right? Think about that. Wow. Divide, and then eventually you have enough cells, and then it's like, oh, we're going to divide these. Those are going to become your heart. Those are going to become your lungs. Those are going to become your toenails, right? All of those things. They all come out of that. And so every stem cell has the capacity, if given the right input, to become any potential tissue in your body. That's why they are theoretically so valuable medicine, right? Because I can take a stem cell from Callie and regrow pancreas for her. I can regrow a lung for her if I give them the right inputs. So then Callie's a diabetic, we just take her pancreas out and give her a new one. Boom, problem solved, fixed. Okay? You need a heart transplant, you need a kidney transplant, you need to fix your spinal cord injury. If you can make stem cells get committed and integrated and do all of those things, then we can treat a lot of those things. Now, satellite cells, because they're muscle stem cells, they're only going to become muscle cells. Okay? But they are basically cells that hang out in a muscle fiber or adjacent to a muscle fiber. And as Brock said, when you get muscle damage, when you need to regenerate, there's been damage in a muscle, you activate those cells and they will start to divide and integrate themselves into your muscle and immediately and quickly sort of help us repair and make muscles grow. Okay, satellite cells are really important for repair when you've damaged a muscle, and they're really, really important to activate them for allowing muscles to hypertrophy. I mentioned them here, and then on Friday we'll come back and we'll talk about the importance of those, because there's some animal models of studying hypertrophy where they take and they zap the satellite cells with radiation and they don't work, and those animals cannot get bigger muscles, right? So some interesting stuff that goes on. Okay, so we've done those. What do I have here? Some important sort of points about the sarcolemma. Okay, the sarcolemma is a plasma membrane. So it looks just like all the other plasma membranes and cells in your body. It's a lipid bilayer. Okay, remember with lipid bilayers, right? You have the hydrophobic tails and the hydrophilic heads, and it lines up and it looks like this. Okay. Then on the outside, there's going to be potentially receptors for different things that are going to be there, and they're going to be coupled right down here with things like G proteins and stuff on the inside. So that stuff goes by your muscle fiber, it binds to the receptor, and we get a signal that's going to come in, and there's going to be a host of other this kind of stuff going all the way through the membrane. Okay. Another really, really, really important point about the sarcolemma, probably even more important than the fact that it has these receptors on it, is it is an excitable membrane, which we will talk about tomorrow. Okay. By being excitable, we mean the sarcolemma can generate and transmit an action potential. So you can make and transmit electricity across the surface of a muscle fiber. This is really important for our ability to quickly send signals to and from muscle fibers. Okay? As I said, it's got receptors so that hormones and neurotransmitters can bind, other molecules can bind and do those. Really, really important. Okay? Really important stuff. The basement membrane is going to sit under the sarcolemma, and it's going to have these proteins and collagen strands, and it's going to fuse with the epimysium and help attach the muscle fiber to tendons, the neuromuscular junctions, and those kinds of things. It plays a big role in the termination of synaptic transmission. And this, much like kind of what you guys were mentioning, this is going to provide some scaffolding for muscle fiber regeneration. Okay. And so it's sort of like this is, as I said, this is the framing in a house or something. And we can build the rest of our muscle fiber around it in there. Okay. So I think I said that backwards. So here's the sarcolemma. Okay. And then here's going to be what we call the basement membrane. Just going to be here. Okay. So we've got some stuff on, it's going to kind of exist in many ways on both sides. Here's going to be sort of, this is the inside of our cell. Here's the outside. So this is going to be the basement membrane complex. Some sits on the inside of the sarcolemma, some sits on the outside. But it's going to provide all of this scaffolding. You guys don't have to know all of the individual little components of the basement. You don't have to know all of these little things. They're going to be sitting. Okay. I do want to introduce, though, that there's this thing called dystrophin, which is going to anchor. Here's this F part of actin. This is an actin filament in the sarcomere. It helps to anchor that to the sarcolemma. And so this dystrophin protein, if you have muscular dystrophy, you have a mutation in your dystrophin protein. So you can't anchor your sarcomeres, the sarcolemma, very well. And so it leads to lots and lots of muscle damage and muscle weakness and things like that. Okay. So that's the idea behind the basement membrane. Okay. I just want you to know what it is and kind of what it does. You don't have to know all of the little individual components. Okay. All right. Let's talk about T2s. My favorite. So a transverse or a T-tubule is an extension of a sarcolemma, of your sarcolemma. Okay? So what that means is, see if I can do this without spilling stuff everywhere. Imagine this is a muscle fiber. Okay, here's our muscle fiber. Now imagine what I'm going to do is I'm going to take, and it's not going to be plastic, it's going to be extendable. I'm going to press from right here down into the fiber. Okay. And so as it goes down into the fiber, basically there's a little pore, and you come here, and there's going to be a tunnel that goes down into the inside of the muscle fiber, where the sarcomeres and everything else are going to be located. And so what happens with the T-tubule is it's essentially just an extension of the sarcolemma. The sarcolemma runs along, there's a tunnel, you go down the sarcolemma, and it's the same lipid bilayer as you go down into the T-tubule. Okay? And so what this is going to do is make another analogy. Anybody a Star Wars fan? Two of y'all? Not much. Let me see if it'll work. This is the exhaust chute on the Death Star that they shoot the thing down into in the very first segment. What it is it's a tunnel. Action potential comes along the membrane, the sarcolemma. It gets to the T-tubule, and then the action potential then gets moved down into the inside of that. Okay, and that's going to let us transmit a signal really, really quickly, and it's going to go down into the inside. The T-tubule connects to the sarcoplasmic reticulum. As Destiny pointed out, the sarcoplasmic reticulum has a bunch of calcium. Technically, the calcium is contained in the lateral sacs of what we call the terminal cisternae of the sarcoplasmic reticulum. But what happens is the SR is going to sit, I think I have a picture on the next slide. The SR sits right here. This is the terminal cisternae next to the T-tubule. Okay? And it makes what we call a triad. You have a T-tubule, and then you have the sarcoplasmic reticulum on either side. And there's a series of receptors that are going to connect the T-tubule to the sarcoplasmic reticulum. Action potential moves down the T-tubule, and then that T-tubule is going to tell the sarcoplasmic reticulum to do something, to release its calcium. Okay? Now, these I do want you to know. These are going to be important receptors. So the interaction of the T-tubule and the sarcoplasmic reticulum. On the T-tubule, there is a receptor called the dihydropyridine, the DHP receptor. Okay. Here's the DHP receptor. This is my T-tubule. This is the membrane of the T-tubule. Okay. DHP receptor sits right here. It is going to be the link between T-tubule and the sarcoplasmic reticulum. Okay? It is what we call voltage sensitive. Who knows what that is? A receptor is voltage sensitive. But we need to do squats. Should I make you send action potentials down your T-tubules? Voltage sensitive.
You want to like change the how cells is changing voltage and it's
Okay.
saying like spectrum power or you're changing the calcium, potassium, and things and it's the voltage thing action potential.
Very good. And a voltage sensitive receptor is one that will open or close when the membrane potential around it experiences an action. And so while you're sitting there at rest, no action potential, the DHP receptor is closed. And I say we're going to get up and do squats. So you do start to do a squat, action potential comes down, it changes the membrane potential here at the DHP receptor, and it opens the receptor. The receptor will change its sort of conformational state. Okay? The DHP receptor, when it opens itself up because of that action potential, it is going to send a signal to what's called the ryanodine receptor. We abbreviate as the RYR receptor. The ryanodine receptor sits here on the edge of the sarcoplasmic reticulum. And if you look at it under a microscope, it looks like a plug you put in your drain, like in your bathtub or in your sink. Okay. And so the DHP receptor signals to the ryanodine receptor, and the plug in the top part sort of pulls itself out, and that lets calcium come out of the sarcoplasmic. Okay. This is the signal, or this is the receptor that's going to signal for the release of calcium from the sarcoplasm. Okay. So action potential down a T-tubule, activate DHP. DHP signals to ryanodine. Ryanodine then opens and allows calcium. That will become of amazing importance tomorrow as we talk about how you're going to get a muscle contraction. Okay. Now, in the sarcoplasmic reticulum, there are a host of other things. There's some other proteins, things that bind onto calcium that are probably a little bit beyond what we need to do in the class. But I do want you to know there's another enzyme that sits here on the sarcoplasmic reticulum. It is a thing called a calcium ATPase. Okay, a calcium ATPase. An enzyme that ends in ATPase. I know what that does. We ask Riley, you'll talk about those in biochemistry.
ATPase.
Huh?
Yeah, the electron transfer.
Okay, well, so what does it do? It says it's a calcium ATPase, an ATPase enzyme. What is it? What's its job, basically? Okay, yeah, you're thinking of ATP synthase or synthetase, which is the end. But this one, ATPase means it breaks, it uses energy, it breaks it apart. So what this one does, the calcium ATPase, which everybody just calls circa for some reason. I don't know where they got that abbreviation. It's called circa. What it does is you release calcium out of the sarcoplasmic reticulum, and then circa acts like a vacuum cleaner. And it sucks all the calcium back out of the sort of space in the muscle fiber back into the sarcoplasmic reticulum. So you release it, and then circa pulls it back in. If circa doesn't work, within a few hours, that cell will undergo apoptosis. High levels of calcium inside of a muscle cell for a long period of time. And by long, I mean a couple of hours. And it will tell that, so the cell will think something is wrong, and it will undergo apoptosis. Okay? So circa is really, really, really important. It's always working, and unfortunately, it uses a shit ton of energy. Right? To be able to keep doing muscle contractions, you have to release calcium, pull it back in, release calcium, pull it back in, release calcium, pull it back in. Okay? And so we use a lot of energy here. Not quite as much as we do in the cross-bridge cycle, but a lot. Okay? A lot. That's going to be one that's going to go on right there. You guys will see, we may get to it today. Different muscle fiber types have different speeds of their circuit pumps, their circuit. That's going to affect how quickly we can bring the calcium back in. And that's going to be an important thing. Okay. Mitochondria, here they are, right? They don't actually look like this, apparently. Who knew? This is an electron micrograph of one, but they're a little flatter. They don't always look like a little jelly bean or something in 3 dimensions. We'll talk more about this individual structure when we do metabolism. We'll talk about each of the outer and the inner membranes and where the Krebs cycle takes place and how we're going to move things, where the electron transport chain is. We'll talk about all of that stuff. But those are going to be individual little organelles. They're going to be within your muscle fiber. They have their own DNA, right? You got it from your mom. We think mitochondria used to be single-celled organisms that existed by themselves, and we sort of put them into our cells, and they've just kind of stayed there. They're really cool. Okay, so here's some pictures of satellite cells and myonuclei. This is what we call an H&E stain on an individual muscle fiber. The little white structures that are going to be there are either nuclei or satellite cells. We can't really tell which ones are which under a light microscope. When you look at it under an electron microscope, nuclei sit on the outside like this. And satellite cells also sit there, but the satellite cells tend to have a double membrane around them. That's the only real way that we can tell them apart. And we'll do more with each of those things when we get to talking about hypertrophy. They're really, really, really important for those things. Okay. So this shows a sort of activation. I'll bring this slide back when we get to talking about hypertrophy. So don't worry about this slide here. But what you're trying to show with all of this is a satellite cell that's been activated. And it's going to try to begin to proliferate and sort of integrate itself to repair what's going on there. We'll talk more about that though when we get into hypertrophy. Okay, so let's look at the myofibrils. We'll get down to the real kind of nitty-gritty at the level of circuit. Okay, we've got our whole muscle, we've got our fascicles, we have our fiber. Let's pull out the little tubes that make up our fibers, which are called myofibrils. Okay, this is a myofiber. An individual myofibril is made up of many, many, many sarcomeres stacked longitudinally end on end. Sarcomere, 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 sarcomere. Okay. We'll see how they get stacked out like this. This is what one individual sarcomere is going to look like. That sarcomere then is made up of multiple, we call thin actin filaments here in blue, and thick myosin filaments in sort of the red and pink. Okay. We'll blow up what these things are going to look like in just a second. Here we go. Here's a cartoon of a single sarcomere. Okay. You don't have to know sort of the M line and the H zone and some of these things, but I do want you to know sort of the orientation and information about each of these individual filaments that are going to be contained in the sarcomere. Depending upon the textbook that you read, they will say the sarcomere is the functional unit of the muscle. This is the place where force is generated and movement begins is at the level of the sarcomere. I could argue that force is generated by these myosin heads on the thick filaments, but we won't split too many hairs with all of this. Okay? So this is where force comes from. This is where movement comes from. It all comes from changes at the level of the sarcomere. Okay. In a sarcomere, it's going to be made up of these thin actin filaments. An actin filament is two strands of an actin molecule is a sphere, and it's one sphere attached to the next one, attached to the next one to make a long string. You take two of those strings and you wind them together, and you get a thin filament. It's called a thin filament because when you look at it under the microscope, it looks thinner. Okay? These are a smaller molecular weight protein than, say, myosin. Okay? So that's what we've got here, actin filament. Here's a myosin filament, a thick filament. Myosin looks a little bit different. I'll show you guys a picture of a blown up of a myosin, but a myosin is basically a molecule that sort of looks like a sperm. But it has these tails that wind together, and it has a dual sort of head molecule that sits on the end, and the head region is the thing that's going to actually change shape and allow for you to generate force. We'll talk about what makes up the tail, what the different regions on the head are going to let us do, because that's going to be different between different muscle fiber types. Okay, but the general idea here is when we get the proper signal that the thick filaments, the myosin heads on the thick filaments will bind to the actin. They bind on. Once they grab on, then they're going to pull from both directions. The ones on this side are going to pull towards the middle or the inline. They're going to pull this direction. The ones over here are going to pull that direction. And that leads to our sarcomere pulling itself into the middle, which means it shortens. And so as it shortens, then your whole muscle is going to get some pull. It may shorten, and then that may pull on a bone and allow you. You have millions of sarcomeres in a given muscle. Okay. And so you're going to turn on millions of these things when you need to generate large amounts of force and get some movement to take place. Here's an electron micrograph, so high resolution microscopy of sarcomeres. Okay. This area right here is called a Z line or a Z disc. This is the end. So you've got one these are sarcomeres stacked up right here in the middle. So they're labeling this as the M line. This is the midpoint of our sarcomeres. This heavy area on the end, as I said, is called the Z line or the Z disc. This is a place that anchors your sarcomeres. And anchors them together. Gives them something to hold on to, like holding on to the wall and then pull with your other hand. They're really going to be. So that's what this is going to look like. Okay. Here's a different version. They've stained it ever so slightly different, but here you're going to have sort of the things that are going to be the thickest. They're going to look the darkest. This is where you've got both actin and myosin together. Here we've got the H band, the I band, the Z line. Don't worry about what those zones are. It's just between here and here, these are sarcomeres. Where it looks really dark, this is where there is an actin filament and a myosin filament lined up over the top of each other, so it looks really dark. Out here, there's just going to be some additional connections. Okay. All that I want you to know from that picture, but I think the pictures look really cool. Okay, one other little piece of this before we get into sort of what all is going to go on. Okay, here's a sarcomere. Take the sarcomere and turn it, guys. So you're seeing it like this, and I'm going to turn it. So now you're looking directly down into the end of it, and it looks like this. Okay, and what we have is this very distinct arrangement, and this is really important. Okay. You can call this what is in essence a hexagon arrangement where we have 6 individual actin filaments that go around in a hexagon around one thick myosin filament. Okay? And so we do this. The reason it's so important is it's going to give every myosin molecule 6 additional actin filaments it can bind to. That it can pull on, try to generate force. And so by having this 3-dimensional arrangement, we can, in essence, pack in as much actin and myosin as possible in this sort of rectangular area. And we only get force when myosin molecules bind to actin. And so by having this particular arrangement, we can maximize our chances that a myosin in action. So this 6 actin around the myosin, that hexagonal orientation is another example of structure imparting our ability to generate, right? Maximizing our ability to generate force. Okay. About 2/3 of the muscle, because it's bigger, about 2/3 of the muscle protein in your body is myosin. About 1/3 is actin. Even though there are more actin filaments, they're much smaller. Their molecular weight is much smaller. So you got lots and lots of both of these things. And so you can imagine, right, that this, see the sort of the arrangement of these tubes are going to kind of show you off where at each one of these levels, there's going to be an actin filament. And then in our thick filament, you will note that we have these myosin head molecules that are going to all come off at different angles to be able to bind to one of the 6 actin filaments that are going to exist. They exist at about, this is like a 120-degree angle. No, is that right? Geometry. I got to think about this. 90, I think it's like one. Okay. All right, let's dig it a little bit farther down on actin and myosin filaments. Okay, again, this is really important because it's going to differ between different fiber groups. Okay. Here is a thick filament. Okay. Here is going to be what is in essence, okay, this is one myosin molecule. And so when you put the filament together, all of this stuff that we call light meromyosin or the tail, those are all going to get stuck in together down here.
and then there's going to be this hinge area. And then out here at the very end, there's going to be the head region. Okay. So basically from the hinge out to the head, we call this heavy meromyosin. Okay. So light meromyosin forms the tail. Heavy is going to form the sort of hinge region out to the head. Okay. Now, in the head, you're going to have this additional arrangement of proteins within the heavy meromyosin. This is where it gets to be just a little bit confusing. Okay. So on the head, there's going to be what is in essence two binding sites. Okay. There's going to be a binding site for actin on this side and on the other side. Okay. They're going to be here. Adjacent to the actin binding site is going to be an enzyme. There's also an ATPase. This enzyme is called the myosin ATPase. Myosin ATPase. All or almost all of the ATP that Claire used when she was doing her jumping jacks up here earlier was being used up by the myosin ATPase in thick filaments and sarcomeres of muscles that she was contracting. So it's at this enzyme, okay, in the head region of the myosin filament where the vast majority of ATP use during exercises. Okay. So you've got binding site on either side, you have ATPase enzyme on either side. Then we have this inner layer of kind of proteins here, which are going to be called light chains. And they are light because they have a small molecular weight. There's light chain 1, 2, and 3. You don't have to know 1, 2, and 3. You don't have to know that there's 3. I just want you to know there is this thing that is called a light chain that forms the inside scaffolding for the ATPase binding site. Then along the outside of all of this, the outside part of the hinge, sort of leading from the hinge into the head, is something called the myosin heavy chain. You will note these are much bigger protein molecules than the light chains. They're bigger, they weigh more, they're called heavy chain. Okay. One of the things that differs between muscle fibers is this heavy chain. There are different versions of this myosin heavy chain, and there are different versions of the myosin ATPase enzyme. Okay. Those differences are genetically determined, and every one of you is going to have a different proportion of your muscle fibers in a given muscle that have, let's just say, the fast twitch or the slow twitch version, heavy chain, and the ATPase, and working on each other. And that's going to have some real important consequences on endurance, sprinting speed, jumping height, and the energy efficiency of a given muscle fiber that you have. Okay, we'll talk a little bit more about that probably tomorrow, which we can get to all the fiber typing stuff today. That's why I'm showing you guys this. Again, differences in the protein structure on a thick filament in a muscle fiber are going to have very, very big consequences on actual exercise performance. It's about an enzyme and heavy chain. Okay, so when we talk about that with the fiber types, this is where the difference is. And you guys will hopefully, because you have to be able to look down at a microscope to see this, understand why it took us almost 100 years to figure out that these things were different between different muscle fibers and between animals and humans. Okay. Questions about that? Light meromyosin, heavy meromyosin, light chain, heavy chain, binding site, ATPase. Okay. All right. Now let's switch over to the thin filament side. Here are actin molecules, they're little spheres. They get bound together and wrap around each other and make this string or filament of actin. Okay. Not that big of a deal. On the actin filament, though, there is a site, there's a structure, a location where the globular head of myosin wants to bind. Okay? It wants to reach over and grab your neighbor on the shoulder all the time. You're always like grabbing. Okay? Don't do that. Okay? But it's going to be there. And so, in order for us to be able to not contract the muscle when we don't want to, we need to keep the myosin from binding. Unless we tell it that it's okay. Okay? Unless we tell it that it's okay. So there's a couple of proteins that are going to be associated with the actin filament that help us regulate. First one is called tropomyosin. Okay? Tropomyosin. It is a long, thin protein that is going to sit and wind itself around the actin filament. It's easier to see it down here. Okay. And what it does, the tropomyosin functions like the little plastic protective thing that we have in all the plug-ins in our house to keep our daughter from like sticking a fork in there. Okay. It sits over the binding site. So when myosin comes up and wants to try to grab onto the actin, it can't do it. It cannot reach the binding. Okay. That's our kind of protection from myosin binding. It's great. The problem is we have to have a way when we need it to move the tropomyosin out of the way so myosin can bind. And we use a different protein to help us do that. And that protein is called troponin. Now, troponin is a big protein and it has 3 subunits. It has what's called troponin T, troponin I, and troponin C. All I want you to know is that there is a thing called troponin, and it exists. Okay? Troponin is going to be, it's going to sit also on the thin filament at these regular intervals. Okay? And what troponin does is the C subunit, what's called the C subunit, the C subunit binds calcium. It grabs onto calcium, which has been released from the sarcoplasmic reticulum. It then will tell the I and the T subunits to change their shape, like a transformer. It changes its shape, and as it moves, it is hooked onto the tropomyosin. But as the troponin twists and moves, it grabs the tropomyosin, pulls it out of the way, and opens up the binding. Okay. Now, calcium has told us to do a conformational change, it's moved the tropomyosin. Now myosin can actually bind to our target. And then it will break off very, very shortly, we hope. All the calcium gets sucked back into the sarcoplasmic reticulum, and all of this goes back to normal. And that's why you don't have a cramp all the time. Okay? And so I think the important thing to take away from all of this is the normal sort of resting state of skeletal muscle physiology is no contraction. Okay? Relaxation, tropomyosin bound over the binding site. So we have to do something to tell it to contract, rather than do something to tell it to stop contracting. Okay? Evolutionarily, that's really good because if we were just doing a contraction all the time, we'd use energy all the time, and it would be much more difficult to then have to like give it a signal to relax and things than vice versa. Okay. Questions about that? My assumption is that you guys have seen most of this, probably not all of the individual sort of areas and regions of the myosin head, but the rest of it you guys probably have covered a little bit. Okay. All right. As I pointed out, to the chagrin of everybody in 3813 with me last semester, maybe even in the, we do it in the fall, I think we did. We will talk about all of the steps that are going on here from your brain all the way ticking down. The steps, the signaling molecules, and how we get all of this from. I thought about contracting to I actually did a contraction. We'll work our way through most of that tomorrow. Okay. All right. So we'll do the cross bridge cycle. talk very briefly about fiber types, I think, when we can, because we go till 3:15. I think we're supposed to. I'm tired. I'm worn out already from all of this. So by Friday, I'll even be able to talk. It'll be great. So when myosin binds actin, this is called a cross bridge. Okay, this is called a cross bridge. Anytime you generate force, anytime you move, anytime you lift weights, anytime you do anything that requires force, the amount of force that you generate is directly proportional to the number of bindings or cross bridges that are formed between myosin and actin. Okay, resting state, troponin, tropomyosin, myosin can't bind, calcium binds, this thing gets pulled out of the way. See it twist, it pulls these, form a cross bridge. All force comes from cross bridge formation. Okay? This is very, very, very key. Once we get a cross bridge, the myosin head is going to try, it is not always successful, but it will try to tilt, okay, and pull the actin along, depending upon its sort of direction. In its line. That pulling or movement of the myosin head is called a power stroke. Okay, it's called a power stroke. Movement comes from the power stroke. Force comes from the attachment in the cross bridge, but movement comes from power stroke. Right? And those are two very distinct events that we'll talk about and kind of the ins and outs of that on Wednesday. Okay? Ins and outs of that Wednesday. But I'll just give you, I guess, a sort of a silly example. Kiara, you want to do me a favor? No. Reach back and push on the wall. Think you can push the wall down? Try. Just try for me. Okay? Kiara had cross bridges, but no power stroke. She didn't move the wall, right? Muscle contraction, it's isometric. You're generating force, but there's not any movement taking place. So you're getting cross bridge, but the external force is so big that you can't actually do a power stroke. Whereas if you pick something up, Kleenex box, I pick it up, right? I'm getting both a cross bridge formation. I get enough force that exceeds what this thing weighs. I get the power strokes, my bicep shortens. That's what's going on and why we're going to separate those two things into two very distinct events. Okay. I'm going to point out two other things here. Okay. I want you to know one of them. I want you to know one of these things because it's going to become really, really important in the next few years. This is a thing from an old textbook. And it's just talking about some other proteins that are associated with thick and thin filaments. So nebulin is a protein that helps provide some structure to the actin filaments. It helps hold it together. Okay. Alpha actinin is a Z-line protein as well as desmin are Z-line proteins. They sit on the ends of the sarcomeres and they help hold things together. If you ever read research papers about looking at protein levels and things in muscle, they oftentimes use this alpha actinin as a control because it shouldn't change or go up and down very often. But the one I want you to know is called titin. Okay? Titin's really, really important. Way more important than we used to think. We figured this out like in the last, like say, 10 years or there. Okay? Titin is one of the largest proteins in your body. It's like albumin, it's ginormous. Okay. It's also somewhat elastic. Okay. And titin helps to anchor thick filaments to the Z line. We used to just think that titin was there, it provided structure, it held everything together, and it provided some elastic resistance, okay, in the sarcomere to kind of overstretch. What we know now is it does more than that. Titin, just like myosin, can be what we call a molecular motor, right? Myosin grabs things and it pulls. It tries to generate movement. Well, we've learned in the last 10 years that titin can also do the same thing. Okay? It's not just there and passive and providing structure. Under some circumstances, it functions in concert with myosin to help us generate extra force and to be able to help pull along in those particular sarcomeres. And so at some point relatively soon, these guys at the University of Toronto are probably going to win a Nobel Prize for identifying this. We thought for the longest time that all muscle contractions and all force came solely from myosin. That's actually not true. I have seen titin under a microscope on a video at a conference. You can see the titin like contract and shorten. And you can see the muscle move. It's pretty wild. Okay. I just want you guys to be aware of what this is. Okay. What it is, what it does, because you're going to see things more so about this as our field evolves in the next 5 to 10 years. Okay. Okay. Let's do the sliding filament theory. You guys remember the cross-bridge cycle? You guys all want to throw things at me? Julia's rolling her eyes at me. See it? She knows. This is really, really important. Okay? Because this is where force production and movement worked in concert with each other. And this is how we figure out where that myosin ATPase is using energy. It's not for what you think. Right? It's not for what you think it is. Here's a sarcomere that's relaxed, no contraction. Okay? One thick filament, one thin filament. You're going to get a signal from calcium, binds to troponin, move tropomyosin, we get a cross bridge. Okay? Cross bridge binds here, right? It is then going to have tilted and it's trying to pull these actin filaments together. And then it's going to break apart and repeat that and pull them even closer together. Okay? That whole process is called a cross bridge cycle. And when you get movement like this, where the actin filaments slide together, slide past each other, that's called a sliding filament theory of contraction. Every time you move, this is what's happening. Okay? This is what's happening. I'm going to have to get myself back to a place where I feel comfortable asking you guys to just memorize some shit. You got to memorize this. I'm sorry. This is really, really important. If we don't get this, then a lot of stuff in metabolism and everything else is not going to make that much sense. Okay? Or why certain contractions use more energy than others or generate more force than others. It all comes back to the cross-bridge cycle. Okay? So we are going to start the cross-bridge cycle up here at the top. Okay? What has just happened? The first part of the cross-bridge cycle is we have a myosin molecule that has just been able to bind to actin. Okay? Resting state myosin has an ADP molecule bound to it and a molecule of inorganic phosphate. This has come from splitting ATP apart. But at rest, it sits with these two things bound on. Okay? So, myosin binds. When it has these two things on, we call that the high energy configuration. Myosin has a strong affinity for actin when it is in high energy conformation, high energy configuration. It binds, okay? It then kicks off the ADP and the inorganic phosphate, and then we get a power stroke, okay? It releases those two molecules, it tilts, it pulls the actin, power stroke, okay? We've gotten force generation, we've gotten movement. Thing we haven't done yet, we actually haven't used any energy. No energy. So what happens then is myosin stays bound, then we bring in a molecule of ATP, and it binds onto the myosin molecule, and the binding of ATP and its coupling by its hydrolysis with the myosin ATPase gets the myosin head to let go of the actin. Okay? It lets go, The myosin ATPase cleaves the ATP into ADP and inorganic phosphate. That results in the myosin head going back to its high energy conformation. This one here, it's done the contraction, it's done its power stroke, it breaks off. This is called the low energy conformation. Low energy has less affinity for actin, and it can't do another power stroke. So ATP comes in, it breaks, myosin ATPase hydrolyzes it. We now have ADP and inorganic phosphate, and it's ready to go again. So the next time we get a calcium signal, the next time there's an available binding site, we'll make a cross bridge again, and we start the whole thing. You use energy in muscle contractions to relax the muscle. You use energy to get myosin to let go of actin. Don't actually use energy to get force. You don't actually use energy to get movement. You use the energy to be able to continue to get movement or to continue to generate force. That becomes really, really, really important. We talk about why certain contractions feel worse, cost us more energy. The cross bridge cycle is why running downhill is easier than running uphill. Why running uphill is worse than running on level ground. Okay, it's all going to come back to this. Okay, it's all going to come back to this. That's why I need you guys to memorize. Okay, we'll stop here. No sense in starting fiber. We'll do fiber types first thing tomorrow. We'll take a quiz first thing tomorrow, and we'll start talking about the nervous system side of the anatomy, and then all of our signaling processes that we're going to get us to Wednesday where we're going to get okay. All right. Awesome. See you guys tomorrow. Yes, Scott. Okay. So for tomorrow, for the quiz tomorrow, I want you to fill out the stuff, the learning objective things from today. Okay, and then start to work on the second one for tomorrow. And I'll do a little bit of, I'll give you a couple of questions from today's lecture and a couple from what we're going to talk about. Okay, and then every day from that, so the quiz will be on material that is ahead, stuff we haven't talked about yet, so I can get you to look at stuff ahead. Okay, all right, good deal. Bye, everybody.
Is Christina gonna be wrong?
It is.
She never got back to me last week.